Recording. Welcome to Chapter 10, which is all about DNA, DNA replication, transcription, translation, viruses, and proviruses and prions. It's going to be really interesting. So, what has the genes? In other words, where do we find genes? This is an easy question, especially by now, since you've reviewed all of your eukaryotic, your Mendelian genetic stuff and the chromosomal inheritance stuff. I said this 100 times. Where are the genes at?
Clothing stores.
They're not at clothing stores, woman. Quiet in the cheap seats. The genes are on the DNA, which is in the nucleus. So, that is a really weird bullet. So in the nucleus is DNA, RNA, and protein. We know that genes are carried on the DNA. So DNA is the unit of inheritance. You get DNA from your parents. But 1900s through the 1940s, nobody knew that. They knew that DNA, RNA, and protein were in the nucleus. However, they did not know which of those three coded for specific traits. They didn't know what was the genome. Was the genome made of DNA? Was the genome made of RNA? Or was it made of protein? Well, let's see how the world found out. So we're going to take ourselves back in time to 1941. And so the first experiment we want to look at was done by Oswald Avery and bacterial transformation. So what is bacterial transformation, you ask? It's when a bacteria grabs new genes. Now, it'll grab new genes from the environment around it or from another bacterial cell. Either way, the bacteria ends up with a gene it didn't have before. So when a bacteria is transformed, it has a new gene. So Oswald Avery isolated bacteria from pneumonia patients, and he found two basic types. The R type, which was the R stood for rough, was a harmless bacterial species. And then the S type was a smooth-looking bacterial cell, and it was pathogenic. In other words, he injected R-type bacteria into mice, and they were just fine and dandy. But when he injected S-type bacteria into mice, hooray, science. So, like I said, mice were injected with R cells, which was harmless, and they lived. Then mice were injected with S cells, which is pathogenic, and so they got pneumonia and died. The next thing he did was he took the S cells, which were dangerous, these were the pathogenic ones, and he heat killed them. In other words, he raised the temperature of the solution the cells were in to the point that the cells boiled alive and died. And so now he had dead S cells. And he injected mice with those heat-killed S cells, and the mice lived. Hooray! So, amazingly enough, when you kill the pathogen, it doesn't cause a disease. But what it does do is leave its sort of heat-killed chunks around. So the next thing he did was he injected heat-killed S cells into mice that had previously been injected with the R cells. Remember, these mice were living just fine and dandy because R cells are harmless. And so he injected heat-killed S cells into mice that were living fine and dandy with their R cells. Something kind of funny happened. Those mice died. Somehow, the harmless R cells were becoming pathogenic. They picked up disease-causing genes from the dead S cells. So here we go. There's the rough strain, and bam, injected, the mouse lives. Here's the smooth strain, injected, mouse dies. Heat-killed smooth strain injected, mouse lives. Rough strain and heat-killed smooth strain injected, mouse dies. So something funky's going on here. And he asked the question, what was causing bacterial transformation? So he went 3 steps further. He extracted DNA, RNA, and protein from the pathogenic S cells. And he made 3 separate extracts. The first extract was protein from S cells. The second extract was RNA from S cells. And the third extract was DNA from S cells. And then he proceeded to inject each extract into mice that had R cells. So when he injected mice with R cells and S cell proteins, the mice lived. So no transformation occurred. The R cells stayed harmless. Then when he injected mice with R cells and pathogenic RNA from the pathogenic S cells, the mice lived. No transformation. So finally, he injected mice with R cells and DNA from the pathogenic S cells. And what do you know? The mice died. So DNA causes transformation. Bum, bum, bum! So that's it. That like closes the book, right? We now knew 1941 that bam, DNA carries genes because only DNA caused transformation. And so the genes for pathogenicity were carried on the DNA. What? Humankind for the win! Except we kind of ignored Oswald Avery's results. So genes from the S cells are in DNA. We kind of ignored that landmark research for 10 years. Well, it could be worse, right? Mendel got his research ignored for close to 100 years. So there's that. All right. So 1941 goes by. Oswald Avery gets his research straight ignored, and everybody's like, what is carrying the genes? I don't know. And Oswald Avery's like, hey, and they're like, shut up, Avery! So then we get to 1952, and we do some experiments with a bacterial virus, a virus that infects bacteria, called a bacteriophage. And this was the Hershey-Chase experiment. Alfred Hershey and Martha Chase. They experimented on bacteriophages. So a bacteriophage is a virus that infects bacteria. Amazingly enough, I think phage is poison and bacteria, or bacterial poison. Anyway, so it infects bacteria. And so it's made of a little shell. So this this complex shell right here looks kind of like an Apollo moon lander, and the shell surrounds and encloses and protects the genes. And the only ingredients in that virus are DNA and protein. So here was something that was going to be useful in determining whether or not DNA or protein carried the genes that cause viral infections. Because the virus alters how genes in the bacteria are read. Meaning the virus hijacks the cell and tells it what to do. Meaning the virus has its own genes. The virus must have its own genes that inform the cell what to do. So a bacteriophage latches onto a bacterial cell and injects its genes, leaving the shell behind. So whatever is injected into the bacteria is carrying the genes. So Hershey and Chase were kind of clever hobbitses here. And they did radio labeling to discover what was injected into the cell and used. So they made DNA glow and they made protein glow. Viruses with glowing protein were allowed to infect the cell, the bacteria. And they found glowing protein outside the cell. Then viruses with glowing DNA infect the cell, and they find glowing DNA inside the cell. So the protein stays outside while the DNA goes inside, meaning it's the DNA that winds up inside the cell, hijacking its instructions and telling it what to do. So there it is. DNA carries the genes. So Hershey and Chase got this one figured out. So between Oswald Avery and Alfred Hershey and Martha Chase, we now knew pretty solidly that DNA carries genes. Hooray! So we knew it was DNA. Now the next big thing was to figure out the structure of DNA. And the first job in figuring out the structure of DNA was to figure out its general shape. And then the second job was to figure out the basic structure. And then the third job, fill in all the details based on that. So, Rosalind Franklin was a genius of a woman, and she took what we called X-ray crystallography photos of DNA. And this allowed for really, really good pictures of DNA. And she figured out that DNA was made of 2 strands, and she figured out that DNA was in a helical shape. So, first job taken care of. Thank you, Rosalind Franklin. We knew it's a double helix. Furthermore, she figured out that the phosphates of DNA were in the backbone. So she even started to break into some of the basic structure. So this is her actual X-ray crystallography photo, in which we know that DNA is a helical shape. So it's pretty amazing that they were able to figure out that it was a helix based on this photo here. So I don't even know the details of their research, how they determined that it was helical based on this photo, because I'm not an X-ray crystallographist. So, but there it is, the actual picture that Rosalind Franklin used to determine that DNA was a double helix and that it had phosphates in the backbone. Next up comes Watson and Crick. Watson and Crick, they came along and they visited merry old England. They were working on DNA too, and the head of Rosalind Franklin's department was like, "Hey dudes, check this out." And he showed them Franklin's pictures and her unpublished upcoming paper talking about the structure of DNA. And he showed it to them without Rosalind Franklin's permission. Whoops. And then Watson and Crick straight used her work to discover that the backbone of the double helix was a sugar phosphate backbone. And they also discovered the base pairing rules: adenine, thymine, guanine, cytosine. So it's not to say they didn't do some amazing things in their own right. I mean, figuring out the sugar phosphate backbone is pretty cool. And then determining the structure of the nitrogenous bases and how they pair up is also pretty cool. It's just kind of sucky that, you know, they kind of sort of stole Rosalind Franklin's work. So, base pairing rules, just to refresh you, since we haven't talked about this since the beginning of class. Adenine joins up with thymine, and guanine joins up with cytosine. Unfortunately, they did not list her as an author on their paper, even though they used a large amount of her work. And they didn't cite any of her work in their paper either. So they kind of just gave her the tiniest footnote on one page of a very long research paper, and pretty much said, this woman did stuff. Who cares? Look at our work. We're amazing. Rosalind Franklin died in 1958, unfortunately. And Watson and Crick got their Nobel Prize in 1962. And the Nobel Committee does not award prizes posthumously. So even though they discovered, the Nobel Committee and the world at large eventually discovered that Watson and Crick did not do it all on their own, that they used Franklin's paper in a large part. She was pretty much robbed of any of the credit she could have gotten for it in life. So, hooray for sexism in the past, I guess. I don't know. It sucks. In fact, Watson to this day won't admit that he used Rosalind Franklin's work. He won't admit that she was helpful at all. But Watson, in his own right, is kind of a nutbag. Because, what was it, about 5-6 years ago, he was being interviewed for a newspaper, and he said something along the lines of, "We don't like to think that all people are born with the same capacity of for intelligence, but people who've worked with black people know that that just isn't true." And then he got all his speaking engagements canceled, and he got his ass fired. I mean, he got himself fired. So, at least they responded to it well. And then later on, he wrote a book in which he addressed that prominently, saying he never had thought of himself as a racist. So, yeah. Scientific titans are never, it's important to remember that even your heroes of science are still people. Like Dawkins is amazing, but he can kind of be a dick. Richard Dawkins, honestly, can be a jerk. Watson was nutty. Francis Crick believed that life was seeded on Earth from space, which actually, considering all things, isn't, we didn't get life itself from space, but it's pretty clear that a number of the ingredients required for life got onto Earth from space. So Francis Crick maybe not that crazy. Neil deGrasse Tyson, I think he likes Star Trek more than Star Wars. So, everyone's got their huge flaws. What?
How is that a flaw?
Star Wars is obviously superior.
It's equal.
Superior.
Equal.
Star Wars had much more action.
Star Trek has, how many different, things to choose from?
One word.
There's TOS, Next Gen, DS9, Voyager, what else?
That is my counter to your argument. Lightsabers. You agree?
Sure.
There it is. Jedi mind tricks. So we now know the structure of DNA. Here's Watson and Crick posing for their photo with their giant model of DNA structure. And here it is. The double helix with the base pairs on there. And so the light yellow is adenine and the green is thymine, and then the red is guanine and the sort of orangey yellow is cytosine. So you can see guanine and cytosine are always paired together. Adenine and thymine always paired together. And what's interesting to note about this DNA double helix is that these nitrogenous bases, adenine and thymine and guanine and cytosine, that hold the helices together, that hold the strands together, those are weak hydrogen bonds. So your DNA itself is held together, both strands are held together by hydrogen bonds, some of the weakest bonds in the universe. So it's just kind of neat to think about that, because if your DNA fell apart, you'd be dead. So it's hydrogen bonds, weak hydrogen bonds, keeping you alive. Woo! So we will go ahead and stop here, and we'll pick up with talking about how cells make DNA.